Hello, my name is Brian, and welcome to Apologetics 101. I am here today again with Donny on our part two of our interview. This time we're going to be discussing junk DNA. Donny Budinsky is a founding member of the Standing for Truth ministry and platform, YouTube channel, so forth. I will put the available links in the description box below. But he has an amazing YouTube channel. I would highly recommend someone subscribing to it if you haven't subscribed to it. He's got incredible content. He does all sorts of content, including theology and so forth. So please subscribe to that channel. But anyway, today my guest is Donny Budinsky from Standing for Truth Ministry, and I can't wait for us to dive into that. This will be for part three of the Tompkins controversy, my newest series in which I am, as a video response series where I'm responding to one Erika of Gutsick Gibbon, but I'm also responding to other critics and so forth that also puts forth their own kinds of criticisms and so forth in the mix of all this. And in any given case, and of course she was here recently. Me and her were going back and forth on a number of things through email, and I guess more on that later. But anyway, today, like I said, we're going to be doing my second interview with Donny concerning this. Except in this one, we focused on junk, what the evolutionists call junk DNA. And I think this is going to be kind of important because that's one of the things that they often cherry pick. The evolutionists think that most of our DNA is non-functional. And this, of course, is false. This has been proven and demonstrated to be false, but it doesn't change the fact that they keep on saying that that's the case. And so a lot of times videos like this have to happen and show that that's not the case. Obviously, Erika's made some comments, both in her response video, and I was just watching her main video the other day for the thousandth time. And in that, she also mentions alleged junk DNA, which she also references very briefly. She doesn't seem to dwell on it as much, but she did talk on it a little bit in her main presentation, and then she also referenced it in her video that responding to my Tompkins interview. And so me and Donny Budinsky from Standing for Truth Ministry tackle some of the points and claims that she makes in that discussion. So without further ado, let's dive in. All right, we're back for part two of me and Donnie's discussion here. Let me share my screen.
Let's continue.
And so they're not designed to just fuse. They're not just hanging out there waiting to fuse.
Do evolutionists think this is a pseudogene that it's inside? And also, what is a pseudogene? You talked about that in your book.
Yeah, so most of the pseudogenes, the so-called pseudogenes, are long non-coding arm in the genome. But most of them now that they have analyzed are functional and important to human health, of health defects in humans. So actually, pseudogenes, or more properly, long non-coding RNA genes.
Okay, you guys, he's going to talk about junk DNA, and I don't care about junk DNA. And the reason I don't care about junk DNA is because one very simple thing disproves the notion that, and I do mean this, like, rarely do you get to say that something like disproves something, but I do feel confident to say that this does in fact disprove the notion that the entire genome is functional, right? Because creationists need the entire genome to be fully functional for their perfect creation genetic entropy stuff. So is the genome fully functional? No, it is not. And the way that we know that it's not fully functional is because of knockout tests. You can knock out megabase portions of the mouse's genome, just get rid of them, knock them out, and they can reproduce and have offspring just fine. And then their offspring can also have offspring. So like if the entire genome was functional and necessary, you wouldn't be able to run any kind of knockout test without some kind of actual consequence, more likely lethal consequence.
Did you want to comment on that, or do you want to go ahead and let her finish?
Yeah, there's a lot to comment on there. So why don't we let her finish her, her full argument, and I'll respond.
All right.
Let's just, okay, so we don't need to talk about junk DNA. We don't need to talk about pseudogenes. We know that the entire genome is not functional. Moving on.
Translated into a protein. I do suspect that a lot of those regions were not.
Okay. Yeah, we can respond.
All right. I've got a couple questions first. I know you've got a lot to say on this.
Yeah.
One is what she said about junk DNA, correct? We have to have the whole junk DNA being functional in order to make genetic entropy work. I know you've done a lot of research and videos on genetic entropy. Is that true or not?
Yeah, we don't require the entire genome to be functional because we do understand that deleterious mutations are accumulating, diseases are accumulating, and so we have degraded from a once perfect genome roughly 6,000 years ago, but because of the fall, mutations have been introduced to the genomes of living organisms. So we wouldn't say 100% functional, but I would argue near functional or really high levels of function, somewhere probably between 70 and 80%. And we do have that kind of evidence. I'll touch on that in a bit, but they require most of the genome being junk or based on evolutionary leftovers because the mutation rate is so high, as you know, Brian. And so they need a genome that's only about 10% functional in order that when these deleterious mutations, these effectively neutral mutations accumulate, to the evolutionists, they're accumulating mostly in junk regions of the genome. So those regions absorb the deleterious effects, and essentially those mutations are no longer harmful. But if the genome is what we say, somewhere between 60 to 85% functional, then that means evolution's impossible because that's somewhere between 60 to 80 deleterious mutations accumulating from generation to generation. That's far too high for large-scale evolution.
Now, the next question I have, I think you're going to be answering that as well, I'm guessing, because that's a big issue, because I know that you brought it up before that they've actually tested it and that they actually require these pseudogenes. But according to her, she said the exact opposite, that they've knocked it out and they're just fine and they can live without it. So we know they don't actually need it, according to her. So I don't know if you want to answer that now or that's just going to be part of what you're going to say here in a little bit.
Great question. I'll answer part of it now and then a little more extensive when I get into some of my slides. Firstly, these evolutionists, they forget the fact or they want to deny the reality that many non-coding RNA genes, like pseudogenes, are only expressed under very specific circumstances or conditions. I engaged Erika in a formal debate, 3 as a matter of fact, going back 2 or 3 years ago. And I've had follow-up discussions with her just randomly in open mics, and I've pointed this out. But yet she still repeats the argument. I've pointed out the fact that a lot of these non-coding RNA genes, again, they're only expressed under certain conditions. Endogenous retroviruses, there's very specific ERVs that are only expressed in embryological development. And so once they're done being useful during those stages, they're shut off. And so if you were to knock that DNA unit out, yeah, there wouldn't be any effect because its job is done, basically. Or if you were to knock out various portions of the non-coding regions of the genome, including pseudogenes, but you were to knock those out when they're not expressed, they're not actually doing anything in that moment, then of course the organism is going to be able to live and thrive. But if you were to knock it out when it's actually expressed, then there would be some kind of negative effect. So that's a very basic point that they seem to miss. You would need unlimited sensitivity to determine if a specific genetic sequence, Brian, was actually junk. Because again, there's so many different scenarios, so many different environmental situations, many different stressors that can turn a gene on. But if you knock that gene out when it's not being expressed or doing anything in that particular situation, because remember, in the laboratory, Brian, as I'm sure you know, they're very strict conditions. They're very narrow. Rather than out in nature and in the wild, organisms are subject to ever-changing environments. And so that's where you need such a high repertoire of DNA elements for adaptive purposes, to act as in redundancy, and so on and so forth. So that's one basic point for now, at least, Brian.
Yeah. Be just like we can live without one of our kidneys, but we can't live with them both.
Mm-hmm.
So under certain conditions, we would have to have the kidneys to function, but
Right.
we can manage to survive with one of them if as long as we have at least one of them. All right. I do have a follow-up real quickly, and that's only
Yeah,
because
no problem.
I don't necessarily have you to be able to answer critics' responses, and I just know that Erika's going to be responding to this. So I'm just curious, when you debated her on this, what was her response to what you had to say on that?
Good question. And typically there's one of two responses. And I can't remember if Erika said this because we debated it so long ago, but I've recently debated Grayson in a formal debate and then also in open mic discussions on junk DNA. And he's basically said, he's acknowledged that yes, some non-coding RNA genes are only active under certain conditions, only expressed under certain conditions, but that's an exception, basically. That is just a few, but the majority are just junk. And so he's underappreciating just how active the genome is. So he wants to argue, and I assume Erika would make the same point. I can't remember in our formal debate, I'll have to go back and look if she actually gave a response similar to Grayson's or if she just denied it altogether. But it's pretty obvious if, and I like to use the example of a car. Take your spare tire, okay? You got 4 tires, your car is moving good, taking you from one place to the next, but you've got a spare tire in the trunk of your car. Well, here's the thing, you don't really need that at the time. Evolutionists would look to that as non-functional redundancy. It's just there, but why is it there? It's an extra copy of tires you already have there that are in use. But if you were to enter an environment where you get a flat tire, well, now that spare tire comes in handy. And so that's what we find in the genome is a lot of redundancy or a lot of these DNA elements that, again, are only going to be expressed or used when required. And so, yeah, that would basically be their rebuttal, but I'm going to show why that's a weak rebuttal.
It's funny that you should mention that, since I had some tire problems recently, and I did have a spare in the back, and that's how I was able to sit there and
right.
fix that problem. So,
and if you didn't have that spare tire, you know, what are you going to be doing? Calling AAA, and it's going to be a real hassle.
I used to have AAA. I don't have it no more. I need to get. Yeah. But all right, go ahead.
Okay, yeah, let me share screen on a couple things. So let's address Gutsick Gibbon here on her claims. That, see, I feel like she kind of hand waved it away. She does the same thing with genetic entropy. Again, I do appreciate Gutsick Gibbon in many ways. I enjoy these technical back and forths with her, and I've had many technical discussions with her. And so, you know, it's important that we're engaging each other, as in the creationists and the evolutionists. So I feel like she hand waved it away, though, because it's a very hard position to maintain in 2023 that the majority of the genome is junk. Back when in 2012, the ENCODE project, they've had many follow-up projects, different kinds of projects, but nonetheless, as you can see here, they discovered tremendous evidence for what's called biochemical function or activity. And so these remarkable findings indicated that upwards of 80%, as you can see on the screen, of the human genome was active to some extent in terms of detectable biochemical activity. They were also surprised to discover that the genome only comprised about 20,000 genes. This was not an anticipated number. We now know that a vast portion of the human genome consists of regulatory elements. They consist of non-coding DNA sequences, which are basically just sequences that don't directly code for protein products. So what people like Erika argue is, I'll go to the next slide here, they'll argue that the majority of this activity is spurious. It's just noise. It's not real beneficial and functional activity. The genome's just really messy, they'll essentially say. Okay, so the question I have on this slide is 80% of the human genome biochemically active? And does this comprise real functional significance? Well, a few reasons suggest that yes. And remember, all of these pseudogenes, all of these ERVs, all of these ALU sequences, these non-coding elements, they are in these regions where we find activity. So if we can show that these regions are functionally active and it's not just spurious, then that overturns all their other claims and assertions, okay? So point number one is, and I'm going to see if I can remember all these, I would say one of the most significant points is if this was really just noise, Brian, if this was mostly just spurious transcription, then evolutionary processes over time, like natural selection, should have weeded out all of this genetic baggage over time, because transcribing portions of the genome generation after generation, replicating this non-functional DNA, would be incredibly wasteful of energy and resources for the cell. Because transcription, right? You got DNA transcribed into RNA translated into a protein. Well, when it comes to a non-coding RNA, you have DNA transcribed into an RNA, and then you have a functional RNA product. Okay, this is actually an energy-intensive process. This is an energy-intensive and resource-heavy process. And so every time you add a single nucleotide, a DNA letter, to a growing RNA chain, you're actually consuming energy. You're consuming ATP. And so if this really was just junk, it's a real basic point, but the evolutionists can't deal with it, and all of this was just noise, then you would think over time, natural selection would find a way to remove all of this genetic baggage. It would be like your drawer or your dresser in your bedroom. If you're just constantly filling it up with new shirts, new shorts, new socks, but you're never throwing anything out, well, eventually your dresser is just going to be overflowing. You're not going to be able to fit anything more. It's going to mess up operations within your room. And so it's good to remove the old as you bring in the new. Okay. Or else it's just going to get too messy, and it's going to be an issue. And so it is with the genome. That tells us that the majority of the activity taking place is actually functional. Another point, transcription factors. What are transcription factors, Brian? Well, they're basically proteins that regulate the transcription of genes. That is, they're copying into RNA, DNA flowing into RNA on the way to make a protein. And these transcription factors, which are found in abundance throughout the genome, they help to ensure that the right genes are expressed in the right cells of the body and expressed at the right time. Now, the point is, the ENCODE project discovered a lot of transcription factor binding. So you have these transcription factors, they're these proteins, they're binding to parts of the genome, and they're helping with transcription. Okay, you've actually got RNA polymerase that can attach to the promoters. You got promoters in the genome. Those are basically genetic switches that initiate transcription. Think of your light. When you enter a room, you turn the light on, right? And so that's your switch to turn the light on. A promoter is a genetic switch to turn on transcription. And so RNA polymerase attaches to these promoters, but only with the help of transcription factors. And the binding sites for transcription factors are often very close to the gene's promoter. And so it all works together. These are interconnected processes. And so if we find a lot of transcription factor binding taking place in the genome, which we do find, that's what the ENCODE project discovered, then the most parsimonious explanation for that, Brian, is what? That it's functional binding. But the evolutionists like Erika, they want to say, oh, this is just random binding. It's just noise. It's not really functional binding. They'll say it's non-functional binding taking place. But if that were the case, and you just had all of this random binding going on and it wasn't functional, well, a couple problems. Problem one, that would just gum up operations in the cell. Secondly, there are only so many copies of transcription factors that are going to be produced. And if these transcription factors were merely binding to random sites in the genome, then there wouldn't even be enough transcription factors available to actually do the binding that is necessary for these transcription factors to properly function. And so in other words, Brian, my brother, there's many good reasons why the transcription factor binding and why the activity in general that we've discovered in the genome is legit function. Okay, so the answer to this question is 80% of the human genome biochemically active? Yes. And does this comprise real functional significance? The answer is yes. Also, Erika is wrong on this claim that they've knocked out genes that in the mice that have had no effect. Yes, that is the case, but there is also the case that they've knocked out non-coding areas of the genome. For example, here's a paper. It's called Multiple Knockout Mouse Models Reveal Long Non-Coding RNAs Are Required for Life and Brain Development. How they concluded this was they actually knocked out various long non-coding RNAs in the mouse, and it was lethal. They died, or some of them survived, but they survived with disease. Okay. So yes, they have actually knocked out various portions of the mouse genome, and it's been lethal. Again, you also have to consider the fact that many non-coding RNA genes are only expressed under certain conditions. There is what's called, and we talked about it a little bit earlier, Brian, redundancy. Okay. So when God created living organisms, the original created kinds, he front-loaded the potential for incredible amounts of adaptability into their genomes. Now, evidence of this comes within the areas of the genome that Erika says are junk or useless. This is redundancy. Engineers know exactly how important redundancy is. We were talking earlier, brother, about how important it is to have a spare tire. Because without that spare tire, you may enter an environment where you get a flat tire and you're going to wish you had that spare tire. So redundancy is basically having backup. And so, yes, we have a lot of backup code, and you could hypothetically knock out genes. And let's say in a mouse, it's going to survive. It'll be just fine because you've knocked out a spare tire. It's got 4 others. Okay. But if a gene or DNA element is mutated or damaged, let's say you get a redundant element, it's mutated, it's damaged, therefore it's used, then the backup genes, okay, they're going to help. They're going to help significantly, but the more you damage these genes, the less backup genes you're going to have. So overall, it's still slightly deleterious because there's only so many spare tires that a genome is going to have. Now, there's another kind of good design that's called over-engineering. When you design or build a bridge, do you build that bridge, Brian, to hold up the exact amount of weight of the exact number of cars that you expect to drive on the bridge? You expect 100 cars to drive on that bridge, so you're going to design the bridge to hold exactly that number, 100 cars? Oh, okay, so then 101 car, an extra car drives over and the bridge collapses. No. Good design is over design. And so you're going to build that bridge 10 to 20 times stronger than the number of vehicles that you expect to be driving on the bridge. Again, this is called over engineering or over designing. And as a matter of fact, it is good design because it adds robustness and reliability. And this is another component of the genome that is there that the evolutionists do not want to acknowledge. So I want to go to a separate PowerPoint here. As I do, Brian, do you have any questions at all on what I've said regarding activity, transcription factors, promoters, anything like that?
No, it seems like very, very good stuff. I can't wait to show this because it's you're right. Essentially, that these genes are designed for a particular purpose, and if the mouse or whatever the organism that's in question is faced with those circumstances, and these genes are not there, then the mouse will die. Is that essentially what you're saying?
Yeah, like the mouse is only going to have so many redundant elements. It's also going to have, as we know, I covered that paper where they did knock out long non-coding RNA genes, and it was lethal. It was or detrimental, where they lived, but they were very diseased.
So it's almost like her saying that that the mouse was okay was kind of misleading. Like that's not the whole story to it.
Right, exactly. So she was right and wrong because, and I see evolutionists do this often, she didn't give the whole story. She should have said, well, they have knocked out various non-coding RNAs, like your long non-coding RNAs, for example, and they've turned out to be lethal. And she could have showed that paper that I've shown, and I'm pretty sure she's aware of it because I brought it up to her in one of our past debates. And then she could follow up by saying, but they've knocked out other areas of the mouse genome and they did not prove to be lethal or harmful. But then I would respond by saying, well, those areas are still active. They're part of that 80% activity, which you're arguing is spurious or just noise. And I'm telling you, no, that can't be noise because transcription is an energy intensive process. And yeah. And
well,
so
well, I was going to say, in her defense, though, she didn't know that you were going to respond to it.
Right. That's true. She didn't know I was going to jump all over this. So we'll give her the benefit of the doubt. But I predict that her response would be, okay, there are some non-coding RNA genes that are only functional under certain circumstances or conditions, but that's just a few. That's an anomaly. That's an exception. But we as creationists would say, no, that's not just an exception. There are many, many, many non-coding RNA genes that are just waiting to be turned on under the right condition or under the right stressor.
Awesome.
Okay. Well, great questions, brother. I appreciate it. So, you can see here, and I'm going to go through, I guess, for sake of making my point stronger, ERVs. Notice increasing knowledge. Brother, this is from 2021. I can show you this type of talk and discussion in papers from 2022, 2023, and in papers discussing pseudogenes, ERVs, ALU sequences, you name it. And what do they say? They say our knowledge about endogenous retroviral elements involved in cell immune responses is constantly what? Decreasing? No, increasing. Because we are in the beginning stages of understanding the DNA language. We are like a kid under the hood of a car looking at the complexity of what's going on, looking at how sophisticated things are under that hood, but not knowing what each part does. It would be like if I was hired to go over to Russia to be a proofreader. Do I speak any Russian? The answer is no. And so am I going to be able to adequately identify what's a mistake and what's not? No. It's going to require a lot of training, a lot of time, energy, and effort to do that job. And that's what's going on a molecular level. Scientists are just beginning to understand the DNA language. If you go back 20 years ago, we as creationists, we didn't really have good answers for why genomes comprise all of this apparent junk, or endogenous retroviruses or pseudogenes. But now in only 20 years, we have dozens and dozens and dozens of papers documenting just how incredibly functional the genome is. Remember, only 2% of the genome is protein coding. So what's the rest of it doing? It's doing a lot of things. It's involved in development. It's involved in redundancy. Again, there's over design, involved in the immune system, involved in all kinds of things, determining cell types. And so we could discuss that all day. And many different types of these long non-coding RNAs, Brian, they've been shown to be involved in gene regulation, protein production, chromosome structure. I mean, we would need probably a good 20 hours straight just to go over the just how functional the genome is. And so biochemical activity is a great way to determine what is and what isn't functional. So there's a lot of slides here, but here's an excerpt from this paper here. Canonical mRNA is the exception rather than the rule. Taken together, these knockout analyses, okay, of these kinds of RNAs indicate that nuclear RNAs are certainly, or non-coding RNAs, are certainly important in embryonic development and disease progression. So we have microRNAs, long non-coding RNAs, all kinds of these non-coding RNAs that are important because they're being knocked out and they're telling us that they're a function. Notice this, because I've heard evolutionists say, oh, it's just easier. Brian, it's just easier for the genome to transcribe all of this junk rather than get rid of it over time. Okay, firstly, the evolutionists want us to believe that through natural selection and mutations and genetic drift and gene flow, recombination, gene conversion, all these processes they look to, they want us to believe that through those processes, you can take a single-celled-like ancestor into a whale over billions of years. You can take your fish to fishermen. You can take your bacteria-like organism to biologists, but you're telling us, evolutionists, that those same processes can't rid the genomes of all this junk? You see how they want to give natural selection unlimited powers in one area, but then in another area, they want to argue that it's weak? And so, yes, clutter is not a good thing. And if most of this activity is just non-functional evolutionary leftovers, that would be a very cluttered genome. Isn't that right, Brian? And so notice, this is all just basic. In any area of life, what does it say? What does it teach us? Clutter can drain all of our energy. There's a number of cons and negative sides to not decluttering your home, not decluttering your house, your car, whatever it is. Okay. And transcription, again, it is an energy intensive process. Every time you add a single nucleotide to a growing RNA chain, you are using energy. You're using ATP. Notice this, ATP is needed for transcription. Energy is also needed when a stop codon stage is reached and the messenger RNA is detached from the DNA. All steps of transcription require energy. ATP is a substance found in all living organisms and is the main source of energy for many metabolic and cell functions, including the transcription of RNA. Okay, so what's the most parsimonious explanation, Brian? If we have a lot of transcription, at least 60% of the genome's transcribed, and there's a lot of transcription factor bindings going on, right? These transcription factors, they're binding to all places of the genome. Then the most parsimonious explanation is that this is functional activity. This is functional transcription factor binding. And to just say, oh, well, it's easier for the genome to replicate all of this junk, that's ridiculous. It's far better to declutter the genome. And there should be processes, if evolution is true, that can do just that.
Hey,
Brian, do you have any questions?
yes, a few. Um, Well, one I was going to ask you, what is jumping genes exactly?
Good question. So you have what's called retrotransposons, like ERVs, for example, would be considered a retrotransposon, but there's various classes. And so many of them can actually jump around the genome. They can move from one chromosomal location to another. And by doing that, they can actually help the genome function. They help with maintaining cell processes through their jumping abilities. And so ALU sequences
So you're
are considered that.
so you're saying that they literally jump wherever they're needed?
Yeah, they have preferred.
That's cool.
Yeah, it's amazing. Yeah, it's, if you notice here, the ALU DNA sequences, which they say are ALU sequences, they will argue that this is parasitic DNA. But notice this paper. Jumping genes have essential biological functions. They're necessary and essential for healthy life processes. The ALU, this is my active research program currently right now. The ALU DNA sequences, which are mobile within our genome, regulate the activity of ribosomes, right? Ribosomes translate a protein, an RNA to a protein, and contribute to the cell's immunity. So without these so-called jumping genes, we would lack immunity. We would lack an immune system, and we'd be riddled with disease from invading viruses. And so there's different types of these transposons. There's LTR transposons, which would be an ERV, because an ERV has LTRs on either side. And then in the middle of the ERV, they got the ENV gag and pull elements. And so, yeah, transposons can literally jump from one chromosome to another, turning genes on, turning genes off. Notice this, non-coding DNA. Look at all these functions, Brian. This is right from the conventional literature, brother. The functions of non-coding DNA. Regulating expression of genes. What's gene expression? It's the process by which a gene is turned on to produce an RNA or a protein. And then you also got non-coding areas of the genome that are regulating that process. So it's all interconnected. Maintaining genome integrity. There's a lot of what's called transcriptionally silent genes, like pseudogenes. And what that means is they lack a promoter. And so they don't produce an RNA. And people like Erika or Dr. Dan Stern-Cardinal, they would say, well, those are definitely junk. Because what are they doing? They don't even produce an RNA. They're not even transcribed. But there's papers suggesting that they are involved in chromosomal stability. As in, if you remove that pseudogene, for example, the genome is negatively affected. It's harmed. The integrity of the chromosome is harmed. And so there's elements there to maintain chromosomal architecture and integrity. So, I mean, you can go down the list and we could discuss each one all day.
Yeah.
And so the point I'm trying to make is there's a lot of function going on, brother.
Right. And one of the things that I was thinking of is that if they really think that junk DNA has no biological function or usefulness, then why don't they just take it all out? They don't do that. So
Right.
So it kind of reminds me of something like a comical thing that Ken Hovind used to do in his preview or presentation, like when he talked about vestigial organs. He said, if you don't think you need your tailbone, I will personally have it removed.
Right. And we should say that to them. If they truly believe that only 10% of their genome's functional, you know what they should do, Brian?
Yeah.
Just knock out the other 90%. Let's see how you feel, right?
Right.
Probably not very good. And we've only knocked out the gold standard of genome functional testings is what? Knockout test. Knock that gene out, snip it out, see what happens. But even that's difficult because, again, as I've pointed out 3 or 4 times, many parts of the genome are only expressed under certain conditions.
Hello, I'm adding this in post-production. And I wanted to add one more thing to what Donnie's saying here. Essentially, in order for Erika to be right and most of the genome to be non-functional, The knockout test can only be applied, has to be applied, be able to apply universally. That means that no matter what the conditions are, no matter which gene they knock out, no matter if 90% of the genome is non-functional, then you should be able to take these non-functional elements, these alleged non-functional elements, and every single time, no matter which one you knocked out, no matter what the conditions were, that it would ultimately produce no effect on the organism. Now, what Donny is saying is essentially no, that's not correct, because yes, there have been knockout tests where they have done that because of whatever situation or maybe the job of the particular gene is done or has already been expressed or whatever the case may be, or there's some redundant elements here. But he's also saying, hey, look, there are experiments, knockout experiments, that has been done where it did cause lethal effects and or diseases to arise or some kind of mutation or some kind of harmful effect on the organism, usually lethal, and that's happened before. So this shows that this is not universal. So Donny is actually demonstrating what he's saying here. Whereas Erika cannot demonstrate it, because even though there are cases where that's happened, there's also cases where that didn't happen. And this is the way science works, right? You just kind of, you should get the same results every time you run the same experiments. If you get different results, you got to figure out what happened that was different. Why did you get different results from the last time you did the experiment? You shouldn't be able to sit there and get different results if your conclusions were sound. You should be getting the same results. And so that's how science operates. And so Erika's saying no, it's kind of cherry picking the data and saying no, there are certain experiments where it doesn't have no effect, but then she doesn't kind of mention all the other times where it did. And so that's problematic. But I wanted to add that in post just to kind of clarify that, because as I'm editing this, I'm anticipating the possibility of arguments that's going to come or responses that's going to come. And I'm sure, and I've dealt with Erika before, and she has a tendency to try to, stick to her guns, even though, she's shooting blanks. So I just wanted to point that out.
2, it's unethical in humans. So if it's happening in humans, it's happening behind closed doors, because it's not ethical to knock out genes in humans. And so we know very little really about what knockout tests could result in, especially in mammals. Okay, so again, we understand very little of the DNA, but we do understand that the majority of the genome is active. And so the question is that activity spurious, or is that activity functional? And do you know what a good way to determine or answer that question is, Brian? It would be by studying the interactions between the systems of the cell. As you know, the genome is not just a linear string of letters, A, T, C, G. No, it's 4-dimensional. It's nested. And there's multiple overlapping codes. The genome is chock full of meta information. And so you have to study interactions between the systems of the cell. We have to look at the interactions that are taking place. We have to look to transcription, which I've talked about. We have to look to transcription factor binding, these proteins that bind to various parts of the DNA to initiate transcription. We have to look to histone binding, histone modifications. We have to look to 3-dimensional interactions. These are all what's called biochemical assays. And when we see activities taking place, we can say, okay, this is function. And that's what we see, is we see a lot of interactions taking place in the cell. And that's why I truly believe it is pseudoscience to make the kinds of claims that Erika is making. And you've heard her say it 2 or 3 times in this last clip. She said the majority of the genome is junk. And so she's trying to say that the majority of this activity is just noise, biochemical noise. But as we can see, Brian, that's not the case.
It seems to me like they treat what they call junk DNA about like the way they do vestigial organs. Every time they don't know about a biological feature, they just assume it doesn't have one. And the fact that they don't know, in fact, that's actually a logical fallacy. If I'm not mistaken, that's ignatum argumentum, argumentum, argument from ignorance. Where they just kind of assume if they don't know it, then they must not have a function. But the fact that we don't know what the function is does not necessarily mean it doesn't have one.
Amen. Amen, brother. It's the vestigial organ argument that's been demolished. 100 years ago, they had what, about 140 or 150 vestigial organs and structures. Now we have 0. They'll argue, for a few: appendix, your tonsils, your goosebumps, they'll argue a few, but they used to have 140 to 150. And now I'd say there's 0. But they're now applying this to the genome, because now there's so much about the genome that we don't understand. And so they can essentially make stuff up and claim, well, this is just junk or that's just junk, because we might not understand currently what those areas of the genome are doing.
Hello, another note added in post-production. I wanted to sit there and comment, or at least stress a little bit more on that. Just because we don't know a particular function does not mean that one day we won't know that function. It doesn't mean it doesn't have one. So this kind of assumption that it must not have a function if you don't know that it has a function, it's just bogus. I mean, think of how they view DNA and genetics, 20, 30 years ago, like Donnie said. I mean, a while back, you could probably make a case like that to people, but nowadays it's like the more and more we discover about the DNA, more we discover about genetics, the more we realize these things do have a function to it. And it's one of those things where, like, a while back, we didn't know that much, and a lot of people think, oh well, there's all this junk and that sort of thing that has no biological function. It turns out that it does have a function, and we discover that over time. And just because we don't know the function today, can you imagine what we might know tomorrow? So, I mean, it's one of those things where you can't just simply assume it has no function because we're not aware of it yet. There's just so much of the genetics that we just don't know.
But you'll find people, informed evolutionists like Dr. Dan Stern-Cardinal, they'll say, well, what's your definition of function? To try and trip you up. Well, here's your best definition of function: biochemical interactions that have consequences for the cell. Now we can determine, okay, we see this activity, and this activity is actually resulting in consequences for the cell. And so we can safely say that this is functional activity. There's another hypothesis. It's an interesting hypothesis. It's called the nucleoskeletal hypothesis, which basically suggests, Brian, that DNA is helping to regulate the volume of the nucleus. But we also understand that a lot of the DNA is serving as a mutational buffer, and a lot of that non-coding DNA is helping the growing embryo, is helping in all these different stages of the developmental process. And to say that all of this is squeezed into just 10% of the genome, is that not just pure imagination, in your opinion, Brian?
Yeah. I think their evolutionary presupposition is regulating their thinking a lot when it comes to stuff like that. And they don't even realize that that's happening. But a lot of what they believe about the genome and about organisms and stuff like that is being regulated by their evolutionary presuppositions about the past, and that guides their thinking on it.
Amen. Amen. So if you were to put yourself in the mindset of the evolutionist, how do you think that they would respond to what appears to be a very logical explanation for this activity? Transcription's an energy intensive, resource intensive process. And so energy, ATP, is being used. So why would we argue that that's just noise? Put yourself in the mindset of the evolutionists. Well, what are they thinking? about this, Brian?
I'm not sure.
You see, so the point is it's intuitive, right? It's intuitive that if there's activity and there's transcription or there's transcription factor binding, then it's intuitive to your normal person that this is functional binding. This is functional activity and not just noise. You'd have to believe in evolution. You'd have to have an evolutionary presupposition to conclude that this is just junk.
The assumption is that it's left over from their evolutionary development, and that's why they assume that it's no biological function to it. And so in their expectation of their evolutionary views, they expect leftover DNA from the evolutionary development that is useless to the organism presently. And so that's
Amen.
what they expect. And then they see, oh, wait a minute, we don't know what this does. So therefore that must be left over from that.
Here's the fatal blow. And I want to reiterate this point. If you have high levels of non-specific binding, again, this goes back to the transcription factors that are binding to various parts of the genome to initiate transcription. If this is just non-specific binding or it's random, like there's binding taking place, the evolutionist says, Brian, and you can watch my impromptu debate with Dr. Dan Stern-Cardinal, and he talks about this. It's just the genome's messy. There's just random binding going on. It's not really doing anything. But here's the thing.
Is
What
that
you're then doing?
Is that Dr. Dan from Creation Myth?
Yes.
Okay.
Yeah. So he's a PhD. And so he's good to engage on this topic because he's at least informed of the evolutionist position. But the problem with that, with Dan's argument, is you're actually then titrating out transcription factors. That means we're not going to be able to get the transcription factor binding that's actually necessary in the genome. And so that kind of random binding that someone like Dr. Dan would talk about, it just gums up the operations within the cell. And that's why I would argue that most of the binding must be serving a purpose, or else these operations in the cell would be extremely messy. It would be, and we know the genome is not like that. Okay. And so I think that's an important point to make to these evolutionists that still want to desperately hold on to junk DNA. And so what I like to do, because all the arguments I've used to refute her on junk DNA, you've probably noticed, they're all from the conventional literature. You know, we're refuting them with their own literature. And then we're also refuting them with logical inferences based on the data that we are reading about in the conventional literature. And they simply can't deal with it because it's their own, they're arguing against their own side, basically.
Yeah, this is very, very good stuff. But yeah, so what she's basically saying on the biological or that this is useless and Tompkins is a geneticist and he should know this.
Right.
And he's not an evolutionary geneticist, so he's not going to assume presuppositions of evolutionists when he argues his position.
Right. Right. Well,
but, yeah, go ahead.
In my opinion, evolutionary theory is hindering our advancement in terms of understanding the genome because they're assuming junk. But we as creationists, we assume what? We assume treasure. And so when we go into the genome looking for treasure, what do we find? We find treasure. The evolutionists, I find this funny, they're accidentally coming across these functional roles. And so they're accidentally finding out that these pseudogenes are functional. They're accidentally finding out that many of these ERVs are functional. Imagine they went in with their millions and millions and millions of dollars of government grants looking for treasure. I predict they'd find a lot more treasure, especially because they're just accidentally coming across these functions. I'm excited to see, because what my goal was for this section, and as you can tell, it was pretty heavy. I wanted to leave no stone unturned on the junk DNA because I'm so sick. Junk DNA is junk science. And so I wanted to throw everything at them from every angle. So I am curious to see how a lot of these critics will respond.
Indeed. As he mentioned, junk DNA is junk science. It's useless because the evolutionists assume that there's no biological function involved, but they don't know it, and then this hinders them actually purposely looking for that design function that it actually does have, and so this hinders their perception further. This has been a good discussion. I can't wait for our next part that will come out. Erika said, so I thought she was going to be releasing the video soon, but apparently she wasn't. She said it could be about another month before she releases it. She's real busy right now. So I'm just going to continue moving forward with the Tompkins controversy series, and I will be responding to Erika because she's supposed to come up with something on Richard Buggs. It's supposed to tie in. She says she has some kind of secondary purpose in mind against Tompkins' his study, and one in which he thinks that me and Carter misunderstood or didn't get or just didn't say anything about, whatever the case may be. And I have a feeling I know which direction she's going into. It's just a really, really powerful hunch, and if it is, then yeah. Well, anyway, we'll deal with that when we get to it. But in case, thank you, and I will see you on the next one. Bye.